Okay, well, good morning. Thank you for coming to spend your next 15 minutes with us before lunch. I realize I stand between you and the food, so hopefully we'll make it worth your while. So for the next few minutes, I'd like to introduce you to what we're doing at CDI and start with maybe who we are. Cellular Dynamics International, a Fujifilm company. First of all, perhaps different from what you might have heard before, a Fujifilm company appended. We are the world's largest producer of iPS cell-derived products. We were founded in 2004 out of the lab of Jamie Thompson at the University of Wisconsin and headquartered in Madison. We're a wholly owned subsidiary of Fujifilm as of April of this year. We have about 170 people, deep expertise in stem cell biology, and perhaps one of the highest concentrations of stem cell biologists on Earth in that one little building. And 800 patents owned or licensed, which I reference this just to let you know that we're very serious about identifying the key steps that you need and the pieces you need of the technology and partnering or developing them on our own to make sure that what we do, we have freedom to operate and our customers have freedom to use the product. So when we were founded in 2004, we started with a set of assumptions that I've crudely tried to represent here in this graph. Our fundamental assumption was that there would be a market for stem cell-derived products, and that first market would be research products. And that following that, at some interval, would be therapeutics. And therapeutics would be bigger, who knows how much bigger. So based on this, there was another underlying assumption, which was if we got good at research, then those skills and that platform would translate to the development and manufacture of therapeutics. If we became the standard for research products, we'd have a leg up on becoming the standard for therapeutics. So that's the underlying assumption. I think it's proved to be a pretty robust set of assumptions now 11 years later. And who we really are from a scientific point of view is we develop and manufacture cells to a tight specification, starting with an iPS cell as the fundamental building block. So why Fujifilm? Very briefly, a very big company, 79,000 employees, $20 billion in revenue. Why would they be interested in a partner like CDI? Well, they have 6 divisions, one of which is healthcare. That healthcare division includes small molecules, antivirals, an antibody business, a contract manufacturing division with vaccines, and this regenerative medicine division, which we are a part of, that includes cell therapy, tissue engineering, and if you were lucky enough to hear Toda-san speak this morning, you heard him reference the JTech products. That's an example of what's inside Fujifilm Regen Med. So, but there's this other concept that all of the engineering expertise inside Fujifilm, the manufacturing sciences, materials, their imaging, all of that expertise can be brought to bear on our sets of problems using our expertise, which is basically at its roots cell biology, and we're really good iPS cell geeks. So those two together have really created what we think is some great intersection for new value. And just some examples of this is only 4 months into our synergy as our merger. And we've got projects in tissue engineering, bioreactor design, automation of some of our production steps, imaging technologies, working on media and components. It's been incredible to interact with so many really fantastic experts inside Fujifilm. So we today have a life sciences portfolio at CDI that includes these products, these eye cells, cardiomyocytes, neurons, endothelial cells, dopaneurons, and hepatocytes. And then all of the products below, which are also available in cryovials. You can order them today. They're 95% pure, high viability. You can plate them with our protocols and media. We know how to manufacture those in the billions. And those are just the ones that we talk about. We have lots of other expertise. We have a partnership with Life on the media like E8 and vitronectin and reprogramming, which we've invented. So we have 12 today. There'll be 5 more the next time you hear us talk, maybe in a year, because we've got an aggressive program to build this part of the business. ISO and GMP backbone for our quality system from the beginning. Every product we manufacture has complete traceability, materials management risk profile. We were serious about this from day one because we knew that we would need it when we became therapeutics, when we moved to therapeutics. So this is an example of a MyCell project where the customer brings in this case, 250 blood samples from patients. These are left ventricular hypertrophy patients. We take the blood samples, make the iPS lines, make the pure cardiomyocytes and cryopreserve them, and then hand back 10 vials of 95% pure cardiomyocytes to the customer. We just finished this project last week, so all 250 donors are done. And that's an example of this true intersection. And you can think of it as an autologous research trial. It's the first step towards an autologous cardiac trial. You have to, but obviously not GMP. So we think that we're the gold standard for research, eye cells. I just referenced our website. There's last check 60, but it's probably 80 now, different papers referencing the technology. And it's really exciting to watch. We're really in this phase of adoption. Two that I'd point out, our eye cell neurons are now an accepted, FDA-approved basis for a test to replace the rodent bioassay in a GMP release test for botulinum therapeutic. And the FDA is evaluating the eye cell cardiomyocytes as a replacement for the human ThoroQT trial. We're one of a group that they're evaluating. But exciting developments, sort of the intersection now of these stem cell products into applied testing, which I think is really exciting. So, but from a therapeutics point of view, we have a focus in 3 different areas that I'll talk to you about today. We don't have time to go into this in detail, so I'll just focus on the first and the third. And really, the first one there in ocular and age-related macular degeneration, that's an RPE, today an RPE project, which I'll talk about. We have a project ongoing in photoreceptors I'm not going to talk about, but in the RP and cone dystrophies. Cardiac program, which I'll just have one slide on. And then in neurodegenerative, our focus on a treatment for Parkinson's disease. So first, though, you have to have an IPS line to start. And the question is, are you starting from an allogeneic paradigm, an autologous paradigm, or something in the middle, a partially matched paradigm, an HLA-matched paradigm? We've chosen to enable all 3 of those, but we think we're the leader, certainly in implementing all 3, and especially in the HLA world. The idea here is that we know solid organ benefits, transplants benefit from HLA matching, and since it's IPS, we can test the donor, find the appropriate genotype, and source it as the basis for the IPS line. So that gives you control over the genotype that you couldn't do in other ways. So if you pick the right HLA homozygous individuals, they match a larger proportion. And so if you build a bank of the right people, you could, in theory, match the U.S. population with a small set. We've been aggressively pursuing this idea since 2012. And an example coverage map there is on the bottom. That's an old model, but it's illustrative of what you can do if you had the correct genotypes. It would take about, let's say, 200 people to cover 95% of the U.S. population. Actually not very scary to us at all as far as IPS lines. I just showed you 95% purity cardiomyocytes from 250 people. So we're pretty good at parallel manufacture. Our phase 1 is completed. We have the first top 5 donors in the U.S., the genotypes for HLA already sourced and reprogrammed. We've made 2 GMP master cell banks from 2 of the 5. We've gone to phase 2 now, which is a comprehensive U.S. bank, which will start in January, and phase 3, which is in the planning stages, which is an attempt now to cover the major population centers around the world. And the proposal now is 50% coverage of the major target areas. And we're exploring personal banking. We've announced, so you can look that up, discussions in a partnership with Cord Blood Registry, which is pretty exciting, and that could be the future intersection of all of this. But our autologous trial for age-related macular degeneration. So we've been contracted by the National Eye Institute. Our role is GMP-compliant reprogramming from a blood sample, blood and skin, development of GMP-compliant RPE manufacturing protocols, and manufacture of the RPEs. And the little schematic down there is, you can think of it as CDI inside. The NEI has the patients. They source the blood and the skin, punch and send it to us. We do the reprogramming, the RPE manufacture, hand it back to them for the preclinical work. So this is working really well. Everything's on schedule, and the NEI expects to file their IND by 2017. So it's the first NIH program in autologous IPS therapy, and it's the first autologous IPS trial, to our knowledge, in the U.S. So we already had clinical grade reprogramming established. We established that back in 2013 when we made our first lines, and we can do it from peripheral blood and skin. So that we already had. What we had to do for them was to develop an RPE manufacturing method. The short summary is, in 9 months, and this is a 3-month manufacturing protocol. So you only have really 3 iterations. It's an impossible task. We eliminated 15 killer stop, no-go reagents and essentially developed a new protocol with a novel purification strategy that I think really makes the best RPEs I have seen, certainly I have seen. And this is an example of 63 runs, 5 operators, 17 different IPS lines from 5 different donors, showing that we can reproducibly, and that's the red line, make the product that you would need to implant. This is at a standard I don't believe anyone has achieved yet, that anyone has published. This is well ahead of anything that I've seen. So, and what's interesting is on the far right, you can see the HLA donors. So the process works quite well for the HLA. All the other runs are the AMD donors from the NEI. So we believe this is going to enable their autologous trial. The NEI will file in 2017. The next is a cardiac program, and I'll just summarize it by saying we've made probably 1 trillion cardiomyocytes and cryopreserved them. It was our first product. We know what we're doing with cardiomyocytes. We have a program here I don't have time to talk about, but I have this little video from Jordan Lancaster at the University of Arizona. If he's here, thank you for allowing us to use this. There he is. So shout out to Jordan, thank you. This is a neat little experiment that he provided to us. He did with our off-the-shelf product. You see in the bottom left there, it's a patch of material that's been seeded with our iCell cardiomyocytes. This is an off-the-shelf product. It beats about once a second. You can pace it about twice per second. And you can see in his publication to the right that when you engraft that into a model, a rodent model, you can see remuscularization of the heart, and you can see improvements in ejection fraction, in systole and diastole. So an exciting example of a collaborator of ours, a customer, that did some really great work. But we have a program here. But the last thing I'll show you is our Parkinson's program. So we know that cell therapy for Parkinson's works. It's been 20 years we've known that it works. Fetal transplants worked. They just weren't reproducible. The quality of the cells varied, the quality of the isolation, the clinics, the methods. There were so many variables that confounded the analysis that the majority of patients failed the trials. But some of them didn't. Some of them improved. What we believe is that the science has been waiting, that patients have been waiting for the right cell in industrial quantity. We think we have that right cell. It's a floor plate-derived midbrain dopaminergic neuron. We believe we're the first to industrialize its manufacture. We make billions of these right now. We can cryopreserve them. We're already at scale. And I'll show you some work using those, some quick animal model studies. The purification protocol works well and will enable autologous. So this is just a quick picture, but some examples of engraftment studies in rodents and in MPT-treated cynomolgus macaques. And what it shows is that the cells not only engraft, but will migrate in the appropriate geography. These are short-term, meaning these are 3-month studies, basically. 3 and 6 months is, I think, what you're seeing there. But the cells maintain their dopaminergic phenotype. And there's, I think, different from anything that you've seen so far, no proliferative potential of these cells. When you have the manufacturing done correctly, and there really are no off-target cells there, they do not proliferate when you put them in. So there's the proliferation stain shows a 0. So, and when you look at a functional study, this is an engraftment study in 6 months in rodents showing amphetamine-induced rotation. So this is a standard model for Parkinsonian tremors and using a rodent. And what your eye should jump to is just that blue line is the control and the colored bars are the treatments. And it just shows that various preparations of the iCell dopamine neurons and at various times are showing a time-dependent improvement in the Parkinsonian tremors. So this is an early example of an experiment. It's still early preclinical work, but pretty exciting. So last slide. We are Cellular Dynamics, a Fujifilm company. Our focus is on both the life sciences and the therapeutics market. Fujifilm has, as a partner now, has really helped us to focus on taking full advantage of that research expertise and those markets. We're building out the catalog, building the commercial team. In cell therapeutics, we have these programs I've mentioned to you. We're building an expanded GMP capacity. We're building a CDMO capacity for partnerships and exploring how that CDMO partnership with the HL lines might work. So thank you for your time, and that's what I'd have to say.